Hi, this is a presentation on neural communications. This is for the College of Science on Human Anatomy and Physiology. For our muscles to work or for hormones to be secreted by the endocrine system, it is important for the neural system to function correctly. Neural communication is accomplished when nerve cells fire or when neurotransmitters are discharged at a short distance to make a specific organ's work. All cells in the body have membrane potential. Membrane potential is when the membrane changes from a positive charge to a negative charge in the membrane. This is due to an unequal amount of sodium and potassium as well as protein ions found in the intercellular and extracellular fluids. It is also due to the plasma membrane's contrasting permeability of these ions. Muscles and nerves are excitable tissue that send electrical impulses when excited. It is these electrical signals that these tissues use to gather, process, start, or transfer information. This makes these electrical signals important to the muscular and nervous system. In this presentation, we will further investigate neurocommunication by understanding graded potential, action potential, refractory period, the structure and function of a single synapse, inhibitory postsynaptic potential, and excitatory postsynaptic potential. Graded potentials are changes that occur in the membrane potential that differentiate in magnitude or strength. They happen when they are triggered by a particular stimulus, which then has the gated ion channels open in a particular area of the membrane. The ion then creates the graded potential to depolarize, meaning that they have to change in the electrical charge, making it a more negative charge inside the membrane than outside. If the trigger is a strong trigger, then the gated potential is bigger. The amount of time spent in gated potential is also dependent on the time the trigger lasts. Because there is loss in the current along the membrane, potassium ions leak out of the membrane, so the magnitude of the graded potential is lost as it travels long distances. Even though graded potentials can only travel a short distance, they are extremely important to the workings of the body. Examples of graded potentials are postsynaptic potential, receptor potentials, end plate potentials, pacemaker potentials, and slow wave potentials. These are all made through excitable cells and can be used to start action potentials or long-distance potentials. Action potentials are short, fast, and have big changes in the membrane potential. This occurs when the inside of the excitable cell becomes more positive inside the cell than the outside environment of the cell. Like graded potential, action potentials have a small region of the excitable cell membrane, but action potentials can travel the whole distance of the membrane. Since they don't diminish like gated potential, they can also signal long distances. Threshold potentials, which are in action potential, have a large depolarization occurrence, making the cell more positive than that of the outside of the cell. This can cause the repolarized membrane to become too polarized, creating an after hyperpolarization where the membrane is even more negative than it usually is before restoring the resting period. With action potential, the quick changes from potential to the threshold to peak and then again to resting is what causes the action potential to fire. The refractory period is when the cell cannot repeat an action potential. There are two types of refractory periods, absolute and relative refractory periods. The absolute refractory period is when the action potential is depolarized and repolarized. The relative refractory period is when the membrane is hyperpolarized. It is the time when the action potential cannot be stimulated regardless of how big the stimuli are or how many times it occurs. The refractory periods are caused when the sodium channel gate is not active due to a blockage. When the gate resets, a stimulus then can occur. The way a single synapse works is that it first starts at the axon terminal, which is the presynaptic neuron. It is where the action potential starts. The synapse finishes in a small bowl called the synaptic knob, with the synaptic vessels that hold particular chemical messengers, neurotransmitters, that have been made and put together by the presynaptic neuron. The synaptic knob and postsynaptic neurons are nearby but do not touch. The open area between them is called the synaptic cleft. It has tentacle-like protrusions that interlock, making that area stable. That way, a synapse can occur. Inhibitory postsynaptic potentials, or IPSP, occur when chloride channels are opened. When the chloride channel opens, chloride flows into the cell, bringing a negative charge to the cell. This makes the cell more negative or hyperpolarized. When the cell is hyperpolarized, the membrane potential is driven away, making the membrane more polarized, which reduces the chance of a neuron's ability to fire an action potential. This creates an ion flow inhibitory change in the postsynaptic neuron. The excitatory postsynaptic potential is an excitatory synapse. It is due to positive ions flowing into the postsynaptic cell. This is because the ligand channels have been opened. This allows for sodium to move into the cell, creating a positive ion charge in the cell. This makes the inside of the membrane not as negative as it was at the resting potential, making it a slightly depolarized postsynaptic neuron. Although it cannot bring it to the threshold, it can have the postsynaptic neuron's membrane closer to the threshold, making the membrane more excitable than it was at rest, making an action potential likely to occur. So to summarize, the neuron communication occurs so that muscle function and hormone secretion can occur. Nerve cells fire through neurotransmitters by discharge, making the organs work. Membrane potential is when the membrane changes from positive to negative charge, sending an impulse to excitable tissue. Gated potentials are changes in the membrane potential. They trigger ion channels to open, creating depolarization. The time spent in gated potential varies, and magnitude loss occurs in long distances. Action potentials are fast, short, and large changes in the membrane potential. They happen when the cell is more positive than the outside environment. Action potential can travel long distances. Threshold potentials are large depolarized occurrences that create hyperpolarization. This helps restore the resting period, causing action potential to fire. The refractory period, the cell cannot repeat the action potential due to the inactive sodium gates. Stimuli occurs after the gates are restored. Inhibitory postsynaptic potentials, IPSP, is due to opened chloride channels, making the cell hyperpolarized, reducing the chance of neurons to fire, an action potential, causing the postsynaptic neuron to have an inhibitory change. The excitatory postsynaptic potential, EPSP, is due to positive ion flow in the postsynaptic cell due to the open ligand channels. With an increased flow of sodium, the membrane is slightly depolarized, making it more excitable than at rest, allowing an action potential likely. This is the conclusion of my presentation. The resources used were Sherwood, 2016, Human Physiology from Cell to Systems through Cengage Learning. Thank you, and I hope you enjoyed the presentation.